Everyone, my name is John V. Bogart, and I am a Gene 32 PLA, so Introductory Genetics. And what I'm going to be talking about today is mitosis. And so, what is mitosis? It is the identical genetic replication in order to make identical cells. And so, identical is super important. It is the process that keeps all of your body cells that need to function the same, functioning the exact same way because it replicates DNA. And so, in order to understand mitosis, we kind of need to understand ploidy. And so, ploidy is the number of chromosome sets. Humans have 23 pairs of chromosomes, and each chromosome number, this is chromosome 1, comes in 2. So that means diploid. Humans are diploid organisms. Di meaning 2, ploid meaning sets. So 2 sets for each chromosome. And so where you get your chromosomes, you get chromosomes from mom and chromosomes from dad. And so these are whole chromosomes, but they can also look like this. Each side is a homologous chromosome, which basically means, similar to this, they just are the exact same size and the exact same shape. And so this is what your cell will look like immediately prior to mitosis. So we have 2n equals 4, 2n meaning diploid, 4 meaning 1, 2, 3, 4, 2 from mom, 2 from dad. These are your 2 chromosomes. They're just split apart right now. And so these chromosomes contain things called alleles. Alleles are your traits that will encode for genetic material. And so we are trying to replicate this cell exactly over and over and over again throughout the course of mitosis, ensuring that you have genetic material from both mom and dad over and over and over again. So, to be clear, there are 2 types of cell division in humans. We have mitosis versus meiosis. The importance is that mitosis is identical replication. So we go from 2n to 2n cells. We just duplicate. Meiosis, on the other hand, is what results in your genetic variation. It is the reason you do not look exactly like a clone of your parents, because genetic variation happens within it. So we go from 1 2n cell to 4 1n cells. The important part is that mitosis is identical replication. And so let's get a little bit more into the process of the cell cycle and mitosis. So it starts off with interphase. Interphase is basically cell growth. It is where the cell prepares for mitosis and gets ready to essentially partake in meiosis. So we have checkpoints G1 and G2. These are just growth checkpoints to make sure the cell is functioning correctly, to make sure it has the nutrients and materials it needs for mitosis. An interphase lasts 23 hours, so it is very, very long because the cell needs to make sure it is ready. S phase is where DNA synthesis occurs. And so in order to create 2 identical cells, 2 new identical cells, we need to replicate our DNA, essentially double it. So you go from this to this. And so this all occurs in interphase, and it occurs in every single cell partaking in mitosis. So once we have a replicated DNA, we can basically confirm the difference between a chromosome and a chromatid. So a chromosome is essentially a piece of DNA that contains genetic material. It can look like this. It can also look like this. A sister chromatid, or a chromatid, is essentially a molecule of DNA. A sister chromatid is the identical half that occurs after replicating the DNA. So let's say we replicated this chromosome in S phase, and now we have another one. Each half of this replicated chromosome is your sister chromatid. And so this will become clear in further examples, but if you have questions about it, please feel free to ask at any point in time. Next, we have M phase. And so this is where mitosis actually occurs. It is the shortest of the phases, about 1 hour, but arguably the most important. So in this we have prophase, metaphase, anaphase, and telophase. And an easy way to remember these is PMAT. It gives you the order of these phases. Prophase is essentially where we have our duplicated chromosomes, so 2 of each. We start to condense. They start to bring together in the middle and just prepare to be replicated. The spindle also develops, which is a structure that helps support the chromosomes. In metaphase, the chromosomes line up at the plate. The metaphase plate is an arbitrary place in the cell. It is not an actual plate. It's just in the middle of the cell. They'll get in line, and these spindles, will start to grow out towards the chromosomes. In anaphase, then our sister chromatids will separate from each other. So this separates from this, this from this, this from this, this from this, and they're being pulled apart by these spindle poles, going this way, each towards the edge of the cell. And finally, in telophase, now our sister chromatids are fully separated. Our spindle is kind of starting to detach because there's no need to pull them anymore. This little nuclear envelope forms to enclose each cell's genetic information. And this envelope forms to create 2 brand new cells. So we started with a cell that looked like this and ended with 2 cells that look like this. And eventually the spindle will go away entirely, leaving room for more replication. And so let's do a quick little recap. We start with a parent cell, 2n, go through prophase, condensing, metaphase, which lines them up, anaphase, which pulls them apart, resulting into 2 daughter cells, with each of them being 2n. They look just like the parents. And quickly, let's do an exercise to make sure we understand the difference between chromosomes and chromatids. So this is what metaphase looks like. This is what anaphase looks like. What is the change in the number of chromosomes, if any, between metaphase and anaphase? So how many chromosomes are in metaphase and how many chromosomes are in anaphase? Feel free to pause here and take a moment to work out the problem. And once you're done, hit play again, and I'll start explaining it now. So in metaphase, we have how many chromosomes? 4. 1, 2, 3, and 4. Now, the difference is, in anaphase, how many do we have? 8 in this case. 1, 2, 3, 4, 5, 6, 7, 8. While yes, these are sister chromatids, they are chromosomes because each of them are individual pieces of DNA. So we have 8 individual pieces of DNA. So this is why sister chromatids can also be called chromosomes, only during anaphase. And so let's look at the number of chromatids and how they change from metaphase to anaphase. So how many chromatids are present in metaphase? 8. We have this sister chromatid, 1, 2, 3, 4, 5, 6, 7, 8. The difference is that they're still attached in the middle by this little portion here. Because chromatids, which is the number of DNA molecules, replicated in the S phase. We went from 4 to 8 in the S phase, and it has stayed at 8 since then. In anaphase, we also stay at 8 sister chromatids. 1, 2, 3, 4, 5, 6, 7, 8. The only difference is that they were pulled apart. This is where the middle used to be. They've just been snatched apart. So I hope that helped with the understanding of mitosis. Please ask any questions as they're needed. And yeah, thank you so much for listening.